Hi, my dear students. We're going to start inflammation. Before you can dive into learning about inflammation, you really have to have a good understanding of the innate immune system. So be sure that you study those slides first. So let's get started by differentiating between infection and inflammation. So infection comes first. So infection refers to a pathogen getting into your body. That can result then in inflammation. So infection first, and then we see inflammation. Now, inflammation is really a response by vascular tissue to some kind of a local injury, which is not only going to occur uh, by pathogens invading us, it could also be because of the presence of damaged cells or all kinds of irritating chemicals, immunosuppression, cancer. I mean, lots of things can trigger inflammation, as you'll see. So we tend to think of inflammation as a not so great thing in the body, but we really need it in order to maintain homeostasis. Now, that's not to say that inflammation cannot get out of hand, and we will talk about that this semester as well. So there are some major functions of the whole inflammatory process. First of all, it's going to make sure that the immune cells and the cells responsible to deal with the inflammation are going to be attracted to the site of injury, and they're going to release all kinds of chemicals. This is then eventually going to lead to tissue repair and eliminate whatever is causing the inflammation, whether it's harmful substances or the pathogens, and even prevent further invasion of those pathogens. So you could argue that that's a fourth function. Now, there are two major types of inflammation. I know that you see three here, but there's really two major types. With this third one, subacute inflammation, is sort of a transition period between acute and chronic inflammation. And of course, by now, I'm sure you realize that uh, the difference between an acute and a chronic process is based on timing or the length of time of the response. So acute inflammation is an immediate response. Chronic inflammation is going to be delayed and keeps going on and on typically as well. So the subacute inflammation is really that phase between, or that transition phase, I should say, between the acute inflammation response and then eventually the full-blown chronic response. And this can take weeks, that transition phase. So as the name says, acute inflammation is going to be a very immediate response to injury. Now, the responses that occur can be occurring locally, but also systemically. So we will talk about these systemic responses as well. It could be as short as minutes or a few days. And very typical for acute inflammation is the formation of an exudate or exudation occurs. So we'll describe what this exudate is all about. In order for acute inflammation to persist or to be sustained, the stimulus, the harmful stimulus, I guess you could argue, needs to persist as well. So once the stimulus is removed, we're going to see that the acute inflammatory process is going to calm down. But again, if the stimulus continues and if the whole process is not properly regulated and gets out of control, we are going to see problems and it could be harmful to the host. All right, so we're going to spend quite a bit of time on acute inflammation, and I'm giving you a bit of an overview here of how we're going to approach the topic of acute inflammation. So in this presentation, we're going to spend quite a bit of time on the cells involved in acute inflammation. It's a quick slide where we list the causes. As well, in this presentation, we will talk about the two major phases that are involved in acute inflammation. There's a vascular phase and then there's a cellular phase. In the next videos, we will talk about the remainder of these items listed here. So here you see a list of pretty easy to remember examples of things that can cause acute inflammation. Notice that even allergens are listed here. Um, and obviously this list is not complete. So the cells involved in acute inflammation can be grouped as resident cells and mobile cells. And when I say cells, they're all going to be white blood cells. And remember, white blood cells can also be leukocytes. Remember that leuko literally means white. So on occasion, I may use the term leukocytes instead. So they're called resident white blood cells because they literally are living in the tissues, typically the connective tissues. Of course, they were created in the red bone marrow via hematopoiesis, but then they left the bloodstream at one point in time and ended up in the tissues. And so those are your resident white blood cells, while the mobile red blood cells are still white blood cells, I meant to say, are still in the blood or still need to be made in the red bone marrow so that they can enter the blood. We'll get to them in just a moment. So it's the resident white blood cells that are responsible for starting the response of acute inflammation. So they do the starting of the whole process because obviously they're the first ones who meet the invaders or the irritants. And we're also going to see that these resident white blood cells are going to have toll-like receptors. Remember that they can also be called pattern recognition receptors. And so these are your most important resident white blood cells that carry um, really good TLRs, meaning that these are going to be receptors that can bind really well to the PAMPs and DAMPs of either damaged cells or pathogens. And so we have our macrophages. Notice the abbreviations I'm going to be using, the dendritic cells, and we'll often mention the Langerhans cells. Remember, they live in the epidermis of our skin. And then we also have the mast cells. Mast cells are very similar to basophils in that they also contain granules filled with heparin and histamine, but they don't have the exact same differentiation, or they didn't follow the same differentiation process, I should say, as the basophils. And then, so what includes the mobile white blood cells? Well, pretty much all of the white blood cells that you know, both the granulocytes and the agranulocytes. And remember that we also have the natural killer cells, which are a type of lymphocyte that is actually granular. Notice too that we list within the agranulocytes the macrophages again. So we have both mobile and resident types of macrophages. And remember, it is the monocytes that form the macrophages. So when monocytes leave the bloodstream, so they are in the blood, and when they enter into the tissues, then they differentiate further into macrophages. So you learned about these different white blood cells in the slides of the innate immune system and even in the adaptive immune system. So I'm not going to revisit all of them. You have that information down already, but we do need to come back a little bit to some of these white blood cells that are going to play a very big role in acute inflammation and some of them in chronic inflammation as well. So both neutrophils and macrophages are important in acute inflammation, but it is the neutrophils that arrive first. So they arrive first, don't forget that. That is very characteristic for acute inflammation. Keep in mind too that both of these, the neutrophils and the macrophages are belong to the professional phagocytes. Keep that in mind. So the macrophages come a little later, and what's interesting is that they are not only going to participate in triggering inflammation, which of course the neutrophils will too, but they're also going to trigger the specific immune system or the adaptive immune system. So we're going to see that the macrophages sort of, well, not sort of, but really have a function in activating both the innate as well as the adaptive immune system in inflammation. Of course, it's the innate immune system that is going to kick in primarily first because your lymphocytes are not going to arrive until much later. I do want to say a few words about how natural killer cells actually function. They have several mechanisms by which they manage to destroy either tumor cells or cancer cells or virus infected cells. Actually, this should say, now that I look at it, this should really say virus infected cells. So what they do is they can either dump their granules or allow for their granules to be released, I should say, which are containing perforin, which drills little holes or creates little holes in the cell membrane of the targets, which are cancer cells, as I said, or virus infected cells of the natural killer cells. And then the granzymes, which are also in the natural killer cells granules, can enter. These granzymes act as proteases. Remember, that means that they are enzymes that break down proteins. And as that happens inside of the host cell, whether it is the cancer cell or the virus infected cell, that is going to draw in water by means of osmosis, and literally the cell will burst or go through lysis. The problem is, if this is a virus infected cell, clearly it's going to also release virions, which are viruses basically. There's not really much of a difference in virions and viruses. It's just that the virions term is often used to refer to the offspring of a virus, of viruses that entered our body. Degranulation can also lead to the release of these molecules that are going to kill various microbes. They're called defensins, more specifically alpha defensins. And the way they destroy bacteria in particular is by literally impacting or destroying the wall of the bacteria. You might recall our discussion on apoptosis with the death receptor, so let's refresh our memory on that. So here on the right in the blue, this would represent the inside or the cytoplasm of the host cell for the natural killer cell. Again, either a cancer cell or a virus infected cell. Here's its cell membrane where where fast receptors are located. And what's going to happen is that the natural killer cell can release a ligand that can bind with the death receptor. And then, of course, that ultimately leads to the caspase enzymes to where apoptosis occurs, killing the host cell. Um, and as I said, degranulation is most often going to lead to the release of perforin and granzyme, to where it then can also cause destruction of the cancer cell or the virus infected cell by means of lysis. Notice that here we do have an antibody involved, and we'll learn how that mechanism works in another topic. So I'm hoping that you recognized here that natural killer cells are kind of interesting because they can go after and destroy cells even if there aren't any antibodies present. I mean, they can also use antibodies, but they don't have to. And they are not dependent on any kinds of major histocompatibility proteins to present antigens. So they have, really fast immune reaction compared to, let's say, the cells of the adaptive immune system, who are very dependent on either an MHC1 or MHC2 protein or even antibodies. Now, one type of cell that we haven't mentioned yet are the microglia. These are cells that we only find in the central nervous system, meaning the brain and the spinal cord. And these are analogous to macrophages, but they are not macrophages. So macrophages are not going to be present in the brain. Microglia are specific cells to the central nervous system. I said brain, but I meant to say central nervous system. And they look like little tarantulas. You see them illustrated here, while the blue ones represent nerve cells with their dendrites and axons. Um, and they can change shape and then start crawling around. Not that we like to think about that, having things crawling around in our brain and spinal cord, but they have the ability, just like macrophages, to recognize and present antigens. And this is important because it's difficult for the rest of our immune cells to get into our brain because there's something called the blood-brain barrier, which is what BBB stands for. It's hard to penetrate that, and antibodies certainly cannot get through the blood-brain barrier. It's also why many medications cannot get into our brain, by the way. It's part of the reason why it's very difficult, for instance, to treat Parkinson's disease, because the medication that could easily treat it just cannot get into the brain all that easily. At any rate, so these microglia go after all kinds of damaged cells or old cells or pathogens. Um, they might clean up plaques. You might have heard of the formation of plaques when um, people have developed full-blown Alzheimer's, etc. They're different cells. They do not originate, we think, in the red bone marrow, like most other immune cells do. Acute inflammation is characterized by very typical signs and symptoms, and they're very often expressed in Latin, but we will also often use the English version of the term. So rubor, tumor, calor, dolor, functio lisa. Actually, I should really pronounce this better, but all of them have a meaning. Rubor literally refers to being red, and when we down the road here, for instance, were to describe this toe, we would not say the toe is red. We would express that as follows. We would say that the toe is erythematous, meaning that it looks red. Think of the word erythrocyte, which is a different way of saying red cell, right? Erythro, red, cyte cell. So erythema refers to red. Tumor, as you know, we use the word tumor in English as well, is some kind of a growth, a swelling, or we will refer to the swelling as edema more often than not. If you speak Spanish, calor makes sense that that refers to heat, dolor, as in referring to pain, and the loss of function, functio lisa, referring to the, clearly it's going to hurt using this toe. Just looking at it gives you the EBDB. So let's reflect on what we've accomplished so far in the topic of acute inflammation. As I tried to tell you in the very beginning of this presentation, the topic of inflammation is rather large, and we're not even halfway through discussing acute inflammation. So we've looked at the various things that can cause acute inflammation, from pathogens to burns, frost bites, to even um, various immune disorders. We've looked at the resident cells versus the mobile cells and learned that the neutrophils are the very first ones to arrive and start the inflammatory process, followed closely by the macrophages. We just also took a look at the very typical classic signs and symptoms of acute inflammation. So we're now going to look at two major phases that occur in acute inflammation, and that is the cellular phase. I'm sorry, that is first the vascular phase, and then we're going to see a much lengthier cellular phase, or more complex cellular phase, I should say. In future videos, we will discuss the following. So the first phase of acute inflammation is called the vascular phase. Clearly, that means that blood vessels are involved, and it goes out like this. So let's say that a tissue is experiencing an invasion of a pathogen, or perhaps there are damaged cells, something along those lines. So that's reflected in this little box that says tissue injury. Initially, that creates a nervous reflex to where the smooth muscle of the blood vessel will begin to constrict, and therefore closing off the vessel to where the blood is not just going to start leaking out all over the place. But that and of course that is going to reduce the blood flow, but that is very short-lived, because then the actual vascular phase continues with vasodilation and an increase in the permeability of the capillaries. Vasodilation is going to expand or dilate the vessels, and that's going to then allow for much more blood to enter the area where there's injury. Um, Therefore, we see redness or erythema. Heat is generated because blood always carries some heat with it. We're going to start seeing, in response to the capillaries getting leaky, we're going to see swelling or edema, and this might also get painful. So here we see yet another big, maybe overwhelming at first, flow chart, but it really is a very informative and very comprehensive diagram that will help you study, and maybe eventually you just need this in front of you to really understand the whole process of acute inflammation. So I'll go over it with you. The red box indicates that this is what we have just discussed. So some kind of tissue injury or perhaps an infection with a pathogen is either going to allow for the DAMPs or the PAMPs. Remember, you learned about those in your innate immune system slides. Those are going to be recognized by the toll-like receptors, sometimes called the pattern recognition receptors, on particularly your mast cells, your dendritic cells, and your macrophages. And as a result, these cells are going to release all kinds of chemical mediators. Now, we will learn about these chemical mediators uh, in future slides here in this particular topic of inflammation. So don't worry about understanding exactly what these do quite yet. You will. So one of the things that will happen initially is that, as we said earlier, first we'll see a very quick neural reflex with vasoconstriction, but then we see vasodilation as well as capillary permeability. We're going to focus here for a moment on just the vasodilation. That is going to cause local hyperemia, literally meaning, let's try to analyze this word because we've learned what emia refers to. Remember, that means blood. So hyperemia means high blood. So lots of blood is arriving at the site of injury due to vasodilation. Clearly, the heat goes up. We see much more redness. Notice that sometimes, again, we use the terms in Latin here, or we can say erythema instead of redness. Of course, if we bring in all this blood, we're also bringing in many more nutrients along with oxygen, which then allows for the cells of the tissue to go through much more cellular respiration and metabolism in general, and that will ultimately lead to healing and resolution, as we will call that process, where the tissue goes back to its homeostatic level. So one of the big characteristics of acute inflammation is the formation of an exudate or exudation. And what exactly is an exudate? Well, when these capillaries get all leaky, then proteins from the blood are literally going to leave the capillaries and end up in the interstitial fluid. So when the proteins, many of them are going to be clotting factors, leak out, they're going to draw out with them water out of the blood, and that's going to then, of course, reduce your blood flow, thicken the blood, and that allows for clotting to start, and then pathogens can get trapped. So let me make a quick drawing so you can imagine this a little bit better. So imagine that here is a tiny little capillary. Of course, I'm drawing it really huge. In our blood, we have all kinds of solutes, but we have quite a few proteins, whether it's albumin, whether it's fibrinogen, whether it's immunoglobulins, um, etc. And of course, we have a whole bunch of water as well. Actually, blood plasma is mostly water, along with all of our blood cells, right? Our white and red blood cells, as well as platelets. But they're really too big to get out of a capillary, even if a capillary becomes very leaky. So let's say that this begins to happen, that the capillaries are getting very leaky, and some of these proteins are going to leak out into the interstitial fluid here to where they're here now. That's also going to draw out a bit of water as well. And so the soup of water plus the proteins, a variety of other things that might leak out of the blood plasma, that's what we refer to as that exudate. Now, as you can imagine, all of this fluid that is accumulating out here, or that exudate, I should say, is going to create some swelling. So edema is occurring, and it may even result in some pain. So now we can focus on the red box that is located more in the center. So the tissue injury, invasion of a pathogen, eventually is going to not just create vasodilation in the vascular phase, but also leaky capillaries to where exudate is formed. That exudate contains proteins, it contains, clotting factors. Honestly, that really should be part of the proteins, because almost all of our clotting factors are proteins. And as you can imagine, when all of these proteins leak out of our blood, it's going to slow our blood, because it's going to lose a lot of those proteins that normally hold in a lot of the water. So the water is going to go into the exudate as well. So some of the clotting factors include fibrinogen that can become activated and form fibrin. We'll learn more about how that works. So this is part of your clotting pathway. And once we have these little strands of fibrin forming, which is kind of how a clot forms, we're going to see that that is going to start the process of healing again. Um, the exudate, as we said, is going to accumulate in the interstitial fluid that creates edema. And this is another way of spelling edema, which means swelling, which might result in pain as well. And that could even impact how well you can use a joint, as we all know very well when we sprain an ankle, for instance. So once that vascular phase has kicked in, the cellular phase can get going as well. And there's a particular order in which the cells are going to become activated, or arrive and then become activated. So first, of course, it makes sense that it is the residential white blood cells that are going to encounter the pathogen or the irritant, or even um, are going to recognize the damaged tissue. And so those residential white blood cells are going to start secreting all kinds of inflammatory chemicals. Some of them are cytokines and chemokines, and we'll learn in more detail what they are. And then once these residential white blood cells are doing their job, they can start attracting, with the help of these chemokines, they can attract other white blood cells. And the first ones to arrive, as I've mentioned earlier, are the neutrophils. So there are two major characteristics in acute inflammation, and that is the formation of an exudate and the fact that neutrophils are the first ones to arrive. So um, remember that we have residential white blood cells, we have mobile white blood cells. So these are mobile neutrophils that are arriving, and they're quickly followed by macrophages and natural killer cells. Now, depending on what kind of an invasion or tissue damage there is, let's say there's some kind of a parasitic infection or perhaps allergies, then we might also see eosinophils. In the case of allergies, possibly basophils arriving as well. And it's not until later on in the process of the cellular phase that we're going to see your T and B cells arrive. But what is important to recognize is that despite the fact that we think of inflammation as more of an innate immune system response, that's not quite accurate. The innate immune system will be the one that kicks in first, but we're going to very quickly see that the adaptive immune system, with the help of your B and T cells, is going to play an important role as well. So next we're going to take a very close look at the detailed steps that the cellular phase goes through. And they are listed here. We're going to see that there's going to be a significant increase in white blood cells called leukocytosis, literally meaning an increase in the number of white blood cells. They're going to be moving towards the site of injury or invasion. They're going to do something called margination along blood vessels, rolling along the edge of the blood vessels, and then stick to the edge of the blood vessels, and then they can actually leave the blood vessels, go through the process of degranulation, release a bunch of their chemicals, and then attract via positive chemotaxis more and more white blood cells. And then we can see the process of phagocytosis occurring. So let's go through this a little slower now. So let's say that there's damage to a blood vessel wall. And remember that capillaries are made up of endothelial cells, or the inside lining of any blood vessel is made up of endothelial cells. They're going to release chemicals, and in this case, these endothelial cells can release something called like leukocytosis inducing factors, and that's going to alert the red bone marrow stem cells to start cranking out more white blood cells, particularly neutrophils, because those are the ones that need to arrive first. RBM standing for red bone marrow. So here on this image, you see a tissue with a blood vessel, a capillary, that is lined with endothelial cell. These are simple squamous epithelial cells right here. Notice the very flattened nucleus. Lots of red blood cells in this capillary, but notice how white blood cells, actually I can tell that that's a neutrophil right there, are finding ways to start lining up along the edge of this blood vessel. So they're getting ready to possibly leave this tiny little capillary to enter into the tissue. So I was already getting a little ahead of myself by talking about the fact that the white blood cells that have left the red bone marrow are now sort of swimming around in the capillaries, um, are starting to get ready to leave the capillary. So they're first going to have to go through the process of margination with rolling and then sticking or getting stuck to the walls of the blood, the inner walls of the blood vessels, to prepare to leave the blood vessels. Now I know that this picture I have here is in German, but it's not important that you understand the words as so much as you're looking at the picture. And by no means do you need to know all of these chemicals that are listed here, right? So, but what you do need to recognize is that in the red here, if you can see red, you might not have a colored picture in front of you, but this is the inside of the blood vessel, the capillary. And here are, or here is a white blood cell. Just a, you pretend it's a neutrophil. And these are your um, squamous cells, your endothelial cells. And so there's going to come a point in time when the white blood cells, let's say the neutrophils that are flowing around in the blood, are going to slowly but surely start getting closer and closer to the endothelial, or to the margin, I should say, of the inside of the blood vessel. So that's the margination, when they're starting to approach the endothelial layer, and then they start to roll along that You can see it here in German, rollen, along the endothelial layer, and eventually they get stuck. They bind, essentially. Um, and so many of the white blood cells or many of the neutrophils will do that, and so then we get ready for them to actually leave the blood. We'll get to that in the next slide. So when white blood cells leave a capillary, there are many ways of referring to it. I like to use the word diapedesis, or sometimes I might use emigration. Notice that it's spelled with the letter E, as in to exit. So they're exiting the blood vessels, not immigrating, but emigrating. And so here, let me kind of fix this picture so that you have a better idea. Here we're looking at a blood vessel lined with simple squamous cells, or I should just say simple squamous epithelial tissue, right? And what they're going to do now is literally squeeze in between these squamous cells to get into our most likely connective tissue. And then they can start to degranulate. So this is clearly a neutrophil because of its multi-lobed nucleus and the little granules. And here you see the degranulation occurring right there. Now, these granules are going to contain all kinds of chemicals that have multiple functions. We'll talk about what complement fragments are. We'll talk more about cytokines, but some of these cytokines are actually chemokines. We'll talk more about them, but they are going to attract more white blood cells towards the site of injury. And that's why we say positive chemotaxis, meaning positive chemotaxis is the term used to say that the cells are moving towards where the stimulus is. Essentially, if they were moving away from it, it would be a form of negative chemotaxis, which is not applicable in our process of inflammation here. So our neutrophils have arrived in the tissues, and again, followed by others, particularly macrophages, but the first ones to arrive are the neutrophils, and they will immediately go after, things like bacteria in particular, maybe some fungi too, and go through the process of phagocytosis. So here we see phagocytosis. You've learned about this before. The bacterium is engulfed in a little vesicle, which we'll call a phagosome. The phagosome then fuses with a lysosome to form a phagolysosome. And remember, the lysosome contains all kinds of pretty harsh enzymes that can digest even organelles if they manage to get to organelles. But here, fortunately, they stay within a vesicle, but together with the poor pathogen. And so the pathogen literally gets digested, and then the phagocyte just spits it back out, all the little particles in the form of what we call soluble debris, and they do that by means of exocytosis. So here we see endocytosis basically at this end. So this is endocytosis at this end, and then we see exocytosis at that end. And so look at that. We have pretty much finished this pretty long flow chart. So now we're focusing on this end of the flow chart where um, the damage to the tissue or the infection by the pathogen triggered the release of all kinds of chemical mediators. Again, we'll get to those, to where vasodilation occurs, increased in capillary permeability, but also leukocytosis, plus a lot of chemotaxis of white blood cells, and first of all, neutrophils. And the neutrophils and the other white blood cells that follow go through a process. So after the red bone marrow has been stimulated to make more and more neutrophils, the neutrophils enter the red bone marrow. They travel, leave the red bone marrow and enter the blood. I meant to say they travel through the blood. That's our migration process. Then they're going to, as they're getting closer to the injury site, they're going to marginate, meaning that they're going to um, start moving towards the edge of the inner blood vessel wall. And then they begin to roll along the inner wall and eventually get stuck. They adhere. And now they are able to literally get through the wall, which is just a simple layer of squamous cells, the endothelial layer of the capillaries. They can pass through that because the endothelial cells have the ability to contract a little bit, and then our neutrophils can slip in between the cells. Um, and how are they going to figure out where exactly to go? Well, the residential cells continue to secrete and release all kinds of chemicals that act as chemotactic, positive chemotactic chemicals, so that the neutrophils will know exactly where to go, and the future neutrophils and the other white blood cells as well. And then eventually our neutrophils run into our famous bacterium or fungus pathogen and engulfs it and then spits it back out as in the form of soluble debris. And we have now healed our tissue essentially. So, what have we done so far in acute inflammation? We looked at the etiology, we looked at the cells involved, all the classical signs and symptoms, and just now we studied in great detail the vascular as well as the cellular phase of acute inflammation.